I'm Fu Tang from Peking University. There are more than 200 types of cells within a human body. Probably one of the most important types of them is human eggs, because they are the only type of cells that can transmit all the genes from a mother to her children and keep the human species immortal. The human eggs are also very special in the way that every egg is genetically different. In fact, this is why every one of us is genetically unique except identical twins. Every egg cell is genetically different, so you have to analyze them one by one to really understand their unique genetics. We recently published a paper in Cell to show that by using cutting-edge technology of mobile single-cell genome amplification and deep sequencing, we can get a better understanding of these mysterious human eggs. Firstly, in a human egg, the two sets of genes coming from grandmother and grandfather need to be rearranged and mixed properly, similar to shuffle of pokers. Every of the gene strings called chromosomes need to be exchanged with its sister chromosome. The rearrangement in eggs is more thorough than that in sperm. We now have a better idea why that happens. Secondly, for the first time, we phase a woman's genome by sequencing her eggs. There are two sets of genes in every cell of a person. It's difficult to separate them to two distinct groups accurately. Now, taking the advantage that within a byproduct cell of an egg, called a second polar body, there is only one set, but not two sets of genes. We can easily separate the two sets of genes of a woman into two distinct groups. In this way, we can get a better understanding of the genetics of the woman.
I'm Dr. Qiao from Peking University Third Hospital. In vitro fertilization is a powerful technology to help infertile couples to get their own babies. The first IVF baby in the world was born in 1978 in the UK, and the first one in China was born in 1988 in our hospital. Although the IVF technology has been successfully used for more than 35 years, the successful rate is still relatively lower, around 30 to 40%. One of the main problems comes from genetic abnormality of human eggs called aneuploidy. It has been shown that when a woman is older than 35 years old, more than 30% of her eggs are genetically abnormal as aneuploidy. These abnormal eggs will lead to either miscarriage or birth of the baby with genetic disease. The real problem is that there is no accurate, efficient, and lower-cost way to detect them. Because of the genetic material within an egg is so tiny that it's too difficult to detect this abnormality with standard molecular biology tools. FISH, SNP, and CGH array have been developed successfully but with limited resolution and accuracy. We analyzed the two polar bodies of one human egg by deep sequencing, simply by counting the number of chromosomes in this byproduct cell as 0, 1, 2, 3, 4. We will know that if the egg is a normal one with two complete sets of genes, or an abnormal one lost or gain some copies of genes. More than this, we can also screen gene mutation in these eggs inherited from the mother. If the mother has some gene copies out of work, we will know that if their out-of-work gene copies are transmitted into these eggs or not. In this way, we kill two birds with one stone. One deep sequencing analysis to avoid two types of genetic problems. We really hope to both help infertile couples to get their own babies in a much more efficient way and also reduce the extra pain of these families by avoiding the transmission of genetic disease from the mother to the baby.
I'm Sunny Xie. Our group has been working on single molecule biology. We published a paper last December reporting our latest single molecule experiment. DNA are single molecules in individual cells. In a human somatic cell, there are 46 chromosomes, 46 individual molecules. Each cell has a different genome containing 3 billion base pairs. If one out of the 3 billion base pairs is a point mutation, we will be able to tell with no false positives. In a sperm or a second polar body from a human oocyte, there are 23 chromosomes. We are able to sequence the genomes of these single cells. To do so, we rely on whole genome amplification. PCR, invented 30 years ago, has single copy sensitivity, but it gives only 6% coverage because it has strong sequence-dependent bias. Some genes are amplified significantly more than others. Multiple displacement amplification, MDA, invented 12 years ago, offers better coverage, but still limited by the sequence-dependent bias. Our new whole genome amplification method, multiple annealing, looping-based amplification cycles, MALBAC, relies on linear amplification, thus providing uniform coverage. Single-cell whole genome amplification by MALBAC is where single molecules meet genomics. Our work showed that the ability to count 1, 2, 3 copies in a single cell is not only now possible, but is critically important as well. I'm very excited about our fruitful collaboration with Jie Qiao and Fuchou Tan on in vitro fertilization. I'm delighted to see an example of a single molecule technology that allows for the probing, understanding, and bettering of life at the single molecule level.